So the process of protein synthesis is going on. As for any other machine, it's working as and when the amino acids are coming in by certain accessory proteins, and these amino acids are joined together by the machine, and these synthesized polypeptides are growing out of the complex, right? So this will continue until a stop codon will come to the A site. Okay, we know that stop codon signals termination of translation, and we know that these stop codons are otherwise also called nonsense codon. Why they are called nonsense? It's because they do not code for any amino acid. That means they doesn't have any sense, right? So all the other 61 codons will code for any one amino acid, but the three stop codons do not code for any amino acid. That's why they are called nonsense codons. And these three stop codons are shown there: UAG, UGA and UAA. They are otherwise also called amber. This is amber, opal and ochre. This is representing their corresponding mutation also. Okay. So moreover, you can also point that there is no tRNA complementary to the stop codon. That's why there is termination is actually happening. So how this occurs? Normally when this stop codon enter the A site, the system or the ribosome will wait for the tRNA to come and occupy the A site. But this will not happen. As we know, there is no tRNA complementary to this stop codon. But all the available tRNAs will come and check whether it is complementary or not, but it will not be possible to interact successfully. So they will not be succeeded. And finally, as additional factor or an external protein called a release factor or termination factors will come and occupy the A site, and this leads to the dissociation of the polypeptide chain from the complex, and this further leads to the dissociation of the entire complex into their corresponding components, right? So there are release factors involved in termination, and in prokaryotes we have three release factors: RF1, RF2 and RF3. And actually this RF1 and RF2 belongs to a particular class of release factor called class 1 release factor, and RF3 is a class 2 release factor. Okay, and these class 1 release factors, that means RF1 or RF2, are responsible for interacting with the codons in the mRNA. That means interaction with the stop codons, right? Whereas the class 2, that means the RF3, is a GTPase protein which is required for the release of the class 1 RF from the complex, right? So when we look at the mechanism, the RF, either RF1 or RF2 will occupy the A site. Then binding of the RF induces the peptidyl transferase. You have to remember the peptidyl transferase activity in the peptide bond formation. And here also the same center of peptidyl transferase activity will transfer the growing polypeptide to a water molecule. Normally what peptidyl transferase do is the growing polypeptide will be transferred to the incoming amino acid or its amino group. That is what the peptide bond. But here there is no amino acid has come, but instead there is a release factor, and release factor do not have any such amino group which is capable of accepting this growing polypeptide. and there is no scope for peptide bond formation. So here what they do is this peptidyltransferase center will catalyze the transfer of this growing polypeptide to a water molecule which is available in plenty in the cytoplasm. So that ultimately causes release of the polypeptide from the ribosome complex. Right. And after that, the condition will be there will be the tRNAs. There will be at least 2 tRNAs, one at E site and one at P site. The mRNA is there. The release factor, that means the class 1 release factor, also will be there in the complex. Now into this complex, this class 2 release factors will come. That means RF3 will come along with the GTP. And due to the GTP hydrolysis, there will be a conformational change in the entire complex, and which causes dissociation of the entire complex into its corresponding components. That means the tRNAs, the mRNA and the ribosomal subunits plus the release factors will be dissociated from the complex. Right. And probably the 30th subunit would have bound by the IF1 and IF3 for the next round of translation. Clear? So this is what's happening during the termination mechanism. Clear? So in brief, there is the stop codon is coming into the A site, then the release factors either RF1 or RF2 will occupy the A site. Then this catalyzes transfer of the polypeptide into a water molecule. As a result, the polypeptide will be released from the complex. Then this is followed by incorporation of the RF3 GTP complex that ultimately causes the complete dissociation of the complex. And this is the overview of that mechanism. Right. You can see that in RF2 you can see certain sequences like GGQ at one end. That means there is a glycine-glutamine sequence at one point at one terminus, and that will be interacting with the peptidyltransferase center actually. And at the other end it has an NIKS sequence. That means asparagine, isoleucine, lysine and serine sequences. This sequence will be interacting with the stop codons. Okay. So ultimately there will be dissociation. So this is happening during the translation event. So now we will also see finally the energy expenditure of translation event. See, for the activation event, we are spending 2 ATP molecules, and for initiation and in initiation, elongation and translocation and termination phases, we are spending 1 high energy compound in the form of GTP. So when you calculate the amount of energy required for a particular number of amino acid, you can calculate it. Right. For example, if you are taking a polypeptide chain of 10 amino acids, we'll have to spend 40 high energy compounds. That means we can generalize it like 4n. So n is the number of amino acids. So this is what the energetics translation. So by this we are coming to an end of translation mechanism in prokaryotes. In the next video, we'll see the mechanism of translation in eukaryotes, how it is different from that of in prokaryotes. So I hope you got an idea about the different mechanisms that we discussed in the videos as 4 sessions. And listen carefully and please ask your queries as a post, and thank you for watching.